Hi, my name is Leonie Luginbuehl. I'm a postdoctoral research fellow at the University of Cambridge at the Department of Plant Sciences. And in today's lecture, I want to give you an overview of beneficial plant-microbe interactions. In particular, I want to talk about two symbiotic interactions, the root nodule symbiosis and the Vascular Mycorrhizal symbiosis. And I want to tell you a little bit about what we understand about the molecular biology that underlies these interactions, but also how we can study these interactions and then use the knowledge and harness it potentially for agriculture. As you know, plants interact with many different microbes in the soil. The soil is full with millions of microbes. There's free-living microbes, bacteria or fungi that can interact with the roots of a plant in one way or another. There's also epiphytes and endophytes. Epiphytes are microbes that can live on the root surface, whereas endophytes are microbes that live inside the roots themselves and sometimes even inside the root cells. And two examples of endophytes are nitrogen-fixing bacteria. These bacteria can live in special organs called the root nodules, and they fix nitrogen for the plant. And another example of an endophyte are the Vascular Mycorrhizal fungi, which again can colonize the roots and live inside the root cells. Now, a lot of these beneficial microbes can have dramatic effects on the plant. They can increase nutrient availability, and that then leads to increased plant growth and can potentially help with crop yield. But they can also help with resistance against biotic and abiotic stresses. And to illustrate these benefits, here's an example of the root nodule symbiosis on the left, where we've got soybean plants that are able and are not able to enter symbiosis with nitrogen-fixing bacteria. So on the left here, we've got plants that cannot establish this symbiosis. And on the right, we've got plants that are able to do this. And you can see very clear differences in plant growth. These ones are much greener and much bigger than the ones that are not establishing this symbiosis. And another example that we have here is an example of the Vascular Mycorrhizal symbiosis in maize. Here we've got the plants that are living with Vascular Mycorrhizal fungi on the left, and these are very green and healthy. And here we've got the same maize plants that are not living with mycorrhizal fungi on the right. And you can see not just differences in growth, but also very clear differences in yield. Now, to exploit the benefits of these interactions, we need to understand the underlying biology and understand more about the partners and the mechanisms that allow the establishment of these symbiotic interactions. And today I want to give you an overview of these two symbioses, because these two symbioses have been particularly well studied and therefore have the potential to be harnessed in agriculture. And most of you will know the root nodule symbiosis. This symbiosis is established mostly by legumes, but the Vascular Mycorrhizal symbiosis is actually much more widespread and much older than the root nodule symbiosis. And I will first give you an overview of the Vascular Mycorrhizal symbiosis, tell you a little bit about the research that I have done with this symbiosis, and then I'll move on to the root nodule symbiosis. And in particular, I want to show you how, even though these two symbiotic interactions look very different on the surface, they're actually very much related. So let's start with the Vascular Mycorrhizal symbiosis. Mycorrhizal fungi in general are fungi that trade soil nutrients for carbon. And there are several different mycorrhizal symbioses, of which the Vascular Mycorrhizal symbiosis is just one kind. So Vascular Mycorrhizal fungi on the left, they associate with most plants. And another big group of mycorrhizal fungi that are related but not identical to the Vascular Mycorrhizal fungi are the ectomycorrhizal fungi. And these ectomycorrhizal fungi only associate with some plants. One of the main differences between the ectomycorrhizal symbiosis and the Vascular Mycorrhizal symbiosis is the way in which the fungal hyphae colonize the cells. In case of the ectomycorrhizal symbiosis, the fungal hyphae that you can see here, they only colonize the outside of the root, and they can grow in between the root cells, but they never actually grow into the root cells. Whereas the endomycorrhizal fungi, of which the vascular mycorrhizal fungi are part, they can actually grow not just on the outside of the roots, but they can grow into the root cells, and then inside these cells they can form these very specialized structures that are called arbuscules. And arbuscules consist of very finely branched fungal hyphae. And because they live inside of these cells, that's why they're called endomycorrhizal fungi as well. Now, the vascular mycorrhizal fungi, they associate with most land plants. More than 80% of all land plant species are able to form this symbiosis. So really, this symbiosis is all around you. And it's established with just a single fungal phylum, the Glomeromycetes. And that's a sister clade to the Ascomycetes and the Basidiomycetes, and only contains about 250 different species. So it's not very many fungi that can establish this symbiosis. And these fungi, they reproduce asexually. They're obligate biotrophs, which means that they're only able to complete their life cycle when they grow in a symbiosis with the plant. And they don't have any host specificity, meaning that one particular fungus can connect with a lot of different plant species, and that then leads to a network underneath our feet, basically, that connects all these different plants with each other. And in addition, this particular symbiosis with a vascular mycorrhizal fungi is ancient. We know that because of some fossil evidence that we have. Here you can see a picture of a 400 million year old fossil, and you can see a plant cell here, and then inside this plant cell we've got this structure that looks a little bit like a tree. And in fact, this structure looks exactly the same as modern arbuscules within plant cells. So these are arbuscules that consist of very finely branched fungal hyphae. And modern arbuscules that you can see here, they look very similar to these 400 million year old arbuscules here. In this case, for the modern arbuscules, what we can do is we can stain the roots with either ink or trypan blue. And that will allow us to visualize these fungal hyphae, because by naked eye, they're not actually visible. So we first have to stain them, and that's one of the experiments that you're going to do as part of this course as well. Now, because of this fossil evidence, today we think that this symbiosis evolved once very early during land plant evolution, about 450 million years ago. This is when plants transitioned from a aquatic environment to a terrestrial environment. And we think that this symbiosis was actually crucial to enable plants to colonize land. And since then, the symbiosis, or the ability to establish this symbiosis, has been maintained by most plant species, with the exception of some plant species that appear to have lost the ability to form this symbiotic interaction. And this includes the family of the Brassicaceae, so including, for example, the model plant Arabidopsis, which cannot establish this symbiosis anymore. Now, in our lab, we study the vascular mycorrhizal symbiosis with the model legume Medicago truncatula, which you can see here. And as in all the other cases, the fungal hyphae, these very thin lines here that you can see, they colonize the roots, and they grow into the roots until they reach the inner cortex, where these arbuscules are formed. And in this case, we have stained the arbuscules, the fungal structures, with a GFP stain, so that's why they appear green. And you can see that these arbuscules here, they have this rectangular shape, and that's because they fill up the entire cortical cell. Now, arbuscules are crucial for the symbiosis because they mediate the main function, which is the nutrient exchange between the plant and the fungus. So in the case of the plant receives mineral nutrients from the fungus. Now, one mineral nutrient that is particularly difficult for plants to take up is insoluble phosphate from the soil. Now, inorganic phosphate often is extremely insoluble, and the plant roots can take it up, but only just in the surrounding of the root. And that then generates a depletion zone, and the roots can't then actually reach phosphate that is further away. The fungal hyphae, however, you can see these very thin fungal hyphae. They're much thinner and they can grow much further into the soil. And they can reach these inorganic phosphate deposits. They can then take them up in the soil and transport them all the way along these fungal hyphae into the roots, where they are released in arbuscules. And it's not just phosphate that vascular mycorrhizal fungi help the plant to take up, but also ammonium and also water. So that leads to a wide range of different mineral nutrients that are being transported from the fungal hyphae to the roots and then provided to the plant. Now, in return for these mineral nutrients, the plant provides the fungus with fixed carbon. The fungus, of course, cannot do photosynthesis, and so it relies entirely on the plant for fixed carbon. And in the case of vascular mycorrhizal fungi, that's particularly important because, as I've mentioned before, they're obligate biotropes, so they rely completely on the plant to complete their life cycle. So arbuscules are the site of nutrient exchange between a plant and AM fungi, and they're particularly well suited to do this. As you can see here, again with these GFP stained fungal hyphae, these arbuscules, here we've got one cell here and one arbuscule here. You can see all these very fine branches, and one purpose of that is to massively increase the surface area through which nutrients can be exchanged. But it's also important to remember that these fungal hyphae, they're not just in direct contact with the cytoplasm of the plant cell, but instead the plants, they form a whole new membrane that we call the periarbuscular membrane that surrounds each of these fungal hyphae. And so this nutrient exchange is very controlled. Colonization of roots by vascular mycorrhizal fungi is a very controlled process, and we can actually define several steps in that process that have to happen in order to have a successful colonization. First, when nutrient availability is very low, particularly when phosphate levels are very low in the soil, plant roots release a hormone called strigolactones. And these strigolactones, they can induce spore germination and hyphal branching, and so can attract the fungal hyphae, the growth of these hyphae, towards the roots. In return, the fungus releases a signaling molecule as well that is called MIC factors, and these MIC factors, they act as symbiotic factors, so they tell the plant that this is a symbiotic fungus, and in particular an vascular mycorrhizal fungus. The fungal hyphae, once they're close enough to the root, then attach to the root and they form these little feet-like structures that we call hyphopodia. You can see them here, and here is an image of these hyphopodia in real life, again here with ink-stayed fungal hyphae in blue. And once the hyphae attach to the epidermis of the roots, they then grow into the root, spread inside the root until they reach the inner cortex where arbuscules are formed. And from the fungal side, this symbiosis is extremely difficult to study because it is an obligate biotroph and because it can't be cultivated without the plant. But on the plant side, in the last 20 years or so, plant genetics has really helped to elucidate the molecular mechanisms of the vascular mycorrhizal symbiosis. And for each of these steps in colonization, there's mutants, plant mutants, that help us to understand how these steps are regulated. For example, we've got wild-type plants. Here you can see an arbuscule up here and other fungal structures. In this part here, you can see a mutant, which is a receptor mutant that can't perceive the fungus. And in this case, you can see that the root is completely empty. All we have is background stain. And that's because this particular plant mutant is not able to establish the symbiosis at all. And we then also have other sets of mutants, for example, this mutant here, which is a metabolic mutant and is not able to form these nice rectangular arbuscules here, but only very small arbuscules. So for each step of colonization, we have identified plant mutants. And one mutant that I am particularly interested in is a transcription factor mutant called RUM1. So a transcription factor, of course, is a protein that regulates the expression of other genes. And this particular mutant here is one that I have characterized further. And what we know about this mutant is that compared to the wild type, again, instead of forming fully developed vascules, the RAM1 mutant that does not have a functional copy of the transcription factor RAM1 only forms these very small abascules here that are not fully functional, and in general the colonization levels are almost entirely absent in this RAM1 mutant. So the question that I had during my research was what is the function of this particular transcription factor RAM1 during AM symbiosis? In particular, I wanted to know which genes are regulated by this transcription factor RAM1 and what is the functions of these genes that are regulated by RAM1. And so to find out more about this, I made use of a hierarchical transcriptional network model that we use generally to think about transcription factors. So I've mentioned that transcription factors regulate the expression of other genes. So if we think about this, if we have, for example, a transcription factor here, then one transcription factor might regulate the expression of a whole number of different genes. And as there might be another transcription factor that regulates a different set of genes. And then some genes might actually be regulated not just by one transcription factor, but by other transcription factors as well. Some of these target genes of these transcription factors might be transcription factors themselves and then have their own target genes. So there's a whole cascade of transcriptional events, a whole hierarchical transcriptional network that we can model for these transcription factors. So what happens if one of these transcription factors is not functional, for example, in a plant mutant? In this case here, if this transcription factor was not functional, what we would expect for these direct target genes here is that they're not expressed, because if it is an activator, then the protein that would activate expression is missing, and so these genes would not be expressed either. And that's true for the genes that are only regulated by this particular transcription factor. But for genes that are regulated by other transcription factors as well, you might expect that you see a reduced expression of these genes. So that's just one example, but I used this kind of thinking to try and find out what the function of RAM1 could be. And indeed, looking at the mutant and at wild type plants, I was then looking for genes in this RAM1 mutant that were not expressed at all, because these could be potential target genes. So to do that, I used techniques that you're learning during this course as well. And in particular, I used global gene expression profiling. So that means I looked at gene expression over the time course of fungal colonization. For that, I extracted the RNA, then synthesized the cDNA, and then sent that to sequencing. So that's different from what you learn in this course, only in the last step, because I'm not doing qPCRs or RT-PCRs to look at just specific genes, but I look at global gene expression. And so for that, I use next generation sequencing. And the way I do that is by looking at 3 different stages of fungal colonization. 8 days after inoculation here, where the symbiosis is just starting, and then at 13 days and 27 days after inoculation. And this time point here represents a fully colonized root. So here the symbiosis is fully established. So then what I do in wild type plants is I compare the gene expression of colonized roots with the gene expression of non-colonized roots, and then see at each time point which genes are either upregulated or downregulated. And for that, I get a number of genes that are mostly upregulated in a wild type, and I then do the same experiment with the RAM1 mutant. And then I particularly look for genes that are not induced at these time points in the mutant background, because these are the most likely targets of RAM1. And the result of that you can see on this slide here, where you can see a heat map that shows the log 10-fold change of gene expressions. So each row is a particular gene, and if it's blue, it means it's not at all induced during a particular time point. but if it's red, it means it's highly induced. And you can see in a wild type that there is a number of genes that are induced at 8 days, more at 13, and the most genes at 27 days after inoculation. But in a RAM1 mutant, there's actually 768 genes that are not induced at all. And these are exactly the genes that we're most interested in, because these are the potential target genes. So what I did next was to then look at these target genes and try to find out what processes they could be involved in. And to do that, I looked at gene ontology terms, which tell us a little bit about the functions of these genes. And what I found was that most of these genes are involved in metabolic processes, and in particular in lipid metabolic processes and fatty acid metabolic processes, but also carbohydrate metabolic processes and glycolysis. But in particular, I got very curious about this lipid biosynthesis that RAM1, the transcription factor, seems to be regulating. Why would lipids be important during this symbiosis? And for a very long time, we did not know that. So to explain that a little bit more, I want to go back to the main function of the symbiosis, which is the nutrient exchange between a plant and the fungus. So I've mentioned before that in return for the mineral nutrients that the plants receive from the fungus, they provide the fungus with fixed carbon. And for a very long time, we thought that this happens in the form of sugars. So here you can see a root cortical cell. And what we thought was that this root cortical cell would export sugars across the perivascular membrane to the fungal hyphae here. And these sugars inside the fungal hyphae would then have to be converted into lipids. This is really important for Vascular Mycorrhizal fungi because they use lipids as the main energy storage form. Indeed, when we look at lipid content in these fungal hyphae and the spores, you can see that there's these white droplets which are lipid droplets, and they completely fill up the spores and the fungal hyphae. And so these lipids are really important for Vascular Mycorrhizal fungi to be able to complete their life cycle. Now, a few years ago, a publication came out that looked at the sequence of the genome of these Vascular Mycorrhizal fungi. And they found that, in fact, the Vascular Mycorrhizal fungi don't have any genes that would allow the conversion of sugars into lipids. So this process does not take place in mycorrhizal fungi. And that then meant that maybe in addition to sugars, the plant also supplies the lipids. And so that's exactly what we found together with collaborators at Rothamsted Research. We found that the plant actually has a whole lipid biosynthetic pathway that will produce lipids from fatty acids that are then exported to these fungal hyphae and that serve the fungus to then be able to complete its life cycle. And RAM1, the transcription factor that I was looking at, turns out to regulate several steps of this lipid biosynthesis pathway. So RAM1 regulates the expression of genes that then encode proteins that will make these lipids for the fungus. And that then explains a lot of the phenotype that we see in a RAM1 mutant, where abascules are not properly formed because this nutrient exchange that includes lipids in addition to sugars does not take place properly. So just recently a paper was published that has taken this research a little bit further, and here they show that actually you can feed Vascular Mycorrhizal fungi with specific fatty acids, and when you do that, then not only do the fungal hyphae grow, but they can also form spores. And that's the first time that researchers can now cultivate these Vascular Mycorrhizal fungi without the plant being present. Because until now, what we didn't know was that the fungus relies completely on the plants for these fatty acids. And now knowing that, we can supply the fungus with these fatty acids and whole new spores can be formed. And this really is a huge opportunity to study the Vascular Mycorrhizal symbiosis a bit more from the fungal side and not just from the plant side, now that we can cultivate these fungi without the plants as well. Right, so we've heard a lot about the Vascular Mycorrhizal symbiosis, and in the last few minutes, I also want to talk a little bit about this other symbiosis, the root nodule symbiosis, which is not established between plants and fungi, but instead is established between plants and nitrogen-fixing bacteria. And macroscopically, these two symbiotic interactions look entirely different. So the mycorrhizal symbiosis, as I've said before, we can't see by naked eye. We have to stain the fungal hyphae with ink or trypan blue. Whereas the root nodule symbiosis you can see just by naked eye, because this root nodule symbiosis involves the formation of these root nodules that you can see here. And these are whole new root organs that you can see. And even though these two symbiotic interactions look very different, they're actually very much related. Now, the mycorrhizal symbiosis is very widespread. I've mentioned before that it's established by more than 80% of all land plant species, but the root nodule symbiosis is very specific to just a few plant families. In particular, legumes can enter a symbiosis with nitrogen-fixing rhizobia, and then there's also some actinorhizal plants that can enter a symbiosis with nitrogen-fixing Frankia bacteria. But unlike the mycorrhizal symbiosis, here the root nodule symbiosis is limited to just a few plant species. Now, the reason why we're so interested in this symbiosis as well is because of the nitrogen fixing abilities of these bacteria. So nitrogen is an essential nutrient for plants. It is the fourth most abundant element in living organisms. It's a component of, for example, amino acids, of nucleotides, but also of chlorophyll, hormones and secondary metabolites. So it's essential together with phosphate as well, that is provided by the avascular mycorrhizal symbiosis. And even though nitrogen is extremely abundant in the atmosphere, so 78% of the atmosphere consists of nitrogen, in this form nitrogen is not available to the plants. So before it is available, it has to be fixed. And this process requires a huge amount of energy. And there's only certain sources of nitrogen fixation. There's lightning that provides enough energy to fix atmospheric nitrogen into ammonia. There's also the synthesis of nitrogen fertilizers through the Haber-Bosch process or fossil fuel combustion. But the biggest part of nitrogen fixation is biological nitrogen fixation. And here we've got marine systems, but also terrestrial systems. And they provide a huge amount of fixed nitrogen per year. And the most sophisticated biological nitrogen fixation is through the symbiotic nitrogen fixation in the root nodule symbiosis. So here again you can see these root nodules, and when you cut these open you can actually stain the bacteria inside these nodules. Here they're stained in blue, and you can see them, these rhizobia now live in these nodules, and they differentiate into bacteroids. And these bacteroids, similar to the mycorrhizal fungi that are not in direct contact with the plant cytoplasm, these bacteroids themselves are as well part of a symbiosome that is surrounded by a specialized plant membrane, which is called the perivascular membrane, the peribacteroid membrane. And the symbiotic nitrogen fixation is extremely efficient compared to other processes of nitrogen fixation. Now, similar to the mycorrhizal symbiosis, we know a lot through plant genetics about the steps in infection and nodule development. And there's two main routes that rhizobia can use to colonize the plant roots. There's the root hair independent entry, which is also called the crack entry. So here we've got cracks in the root surface, and that's where these rhizobial bacteria can enter. And then the cortical cells start cell divisions, and nodules are being formed, and these rhizobia can then live inside the cells in a nodule. But the more sophisticated way for rhizobia to colonize the roots is through root hair dependent entry. So what happens here is that the roots release signaling molecules called flavonoids, And these flavonoids, they are sensed by the rhizobia and attract the rhizobia. The rhizobia in turn produce signaling molecules called nod factors or nodulation factors, which are very similar to the MIG factors in vascular mycorrhizal symbiosis. And these nod factors are then perceived by the plant roots. And what they do is they make the root hair curl and entrap the rhizobial bacteria in this curl. And then an infection thread is formed in the root hairs through which the rhizobia can travel. And at the same time, the cortical cell divisions happen to form a nodule. And once the bacteria then reach these nodules, they're released into the cells and they form these symbiosomes that are surrounded by the membrane. And that's where they can then start to fix nitrogen, which is being provided to the plant. And of course, in return, the plant again provides these rhizobia with fixed carbon from photosynthesis. Now, as in the mycorrhizal symbiosis, plant genetics has helped to understand how the root nodule symbiosis is established. For example, here we've got a wild type root with these very nice pink nodules here. And here is the Cyclops mutant. And you can see that these nodules here, they're very small and not fully formed, and they're white as well. And here is another mutant, the CSIR-K mutant, again, that doesn't form any nodules at all compared to the wild type. And these two mutants are signaling mutants that cannot perceive these nod factors. But what is really surprising is that these two mutants and many other mutants actually not only are affected in the root nodule symbiosis, but also in the vascular mycorrhizal symbiosis. So here, for example, you've got the same Cyclops mutant that cannot form proper nodules. And you can see that even though the fungal hyphae can attach to the root surface, the vascular mycorrhizal fungi also are not able to fully colonize this particular mutant. And the same is true for the CSIR-K mutant. So it seems that we have a lot of the same genes that are required not only for one of these symbiotic interactions, but for both of them. And what we now think is happening is that the root nodule symbiosis actually evolved by recruiting the symbiotic program required for AM symbiosis. So before I've mentioned that the vascular mycorrhizal symbiosis evolved very early on during land plant evolution, about 450 million years ago, whereas the rhizobial symbiosis, the root nodule symbiosis, evolved much later than that, so only about 60 million years ago. And we now think that in the plants that were already able to establish the mycorrhizal symbiosis, all these mechanisms for the establishment of the symbiosis were already present and were then recruited by the rhizobia to start a very similar infection process. So even though the two symbiotic interactions look very different on the surface and the outcomes are very different, the plant program that is required, the molecular mechanisms that are required, are very similar. So to summarize, both of these symbioses have a huge potential to be harnessed for agriculture. The mycorrhizal symbiosis, being so widespread, could help a lot of different crops in taking up nutrients, in particular phosphate, that are already present in the soil much more efficiently. And the root nodule symbiosis essentially produces its own fertilizer by fixing nitrogen from the atmosphere extremely efficiently. Now, one of the caveats of these beneficial interactions, and these two symbiotic interactions in particular, is that they're only established when the nutrient levels are very low in the soil. At the moment in agriculture, we often use very high levels of fertilizers to support plant growth and to increase the yield. And that means that actually these symbiotic interactions are not required by the plant and they're not established. So one of the challenges for the future research is to try and find out how can we optimize fertilizer application so that we have low enough levels that we can still use these beneficial interactions to make nutrient uptake much more efficient. And this would then also help to mitigate the environmental impact of these fertilizers. So to conclude, I just want to briefly summarize what I talked about today about beneficial plant-microbe interactions. We know that symbiotic microorganisms are intimate allies of plants. And these symbioses are mutualistic associations, and that means that both partners benefit. In the case of the Vascular Mycorrhizal symbiosis, mineral nutrients, particularly phosphate and ammonium, from the soil are traded for fixed carbon in the form of sugars, and now we know also in the form of lipids. Whereas in the root nodule symbiosis, fixed nitrogen is traded for fixed carbon. And we've also seen through studies using plant genetics that the plant uses a core symbiotic program for both symbioses, the Vascular Mycorrhizal symbiosis and the root nodule symbiosis, as mutations in this core program affect both symbioses. And finally, we think that the AM symbiosis evolved very early on during land plant evolution, and that the later pathways, for example, in the root nodule symbiosis, are derived from this early AM symbiosis pathway. And with that, I'd like to thank you for your attention. I'll be available for questions, I think, after this lecture as well. But also, if you have any questions, feel free to write an email if there's anything that you would like to know more about, or if anything was not clear. Thank you very much.